Hello, everyone. I would just like to welcome you all today to ASCUS's 2021 Edinburgh Science Festival and to this, our brand new online program, which we launched this year. So this evening, we're presenting Entanglements of Time and Tide, which is an art science engagement with the North Sea and its tidal zones, and it is by India-based artist Sonia Mehra Chawla, who is joining us this evening. So this is a partnership project with Edinburgh Printmakers and Marine Scotland, and we were delighted to host here today this event, which screens an excerpt of The Non-Human Touch, which stems from an intensive international art science residency in ASCUS Lab with Sonia and a long-term collaboration with her over the last few years. So part of this work, the idea behind it is to highlight lab-based science and artistic approaches as a lens through which to explore and investigate the interconnectedness of all living things, both human and non-human. So we were delighted that Sonia's joined us today. After this intro, we're just going to play an excerpt of the film, The Non-Human Touch. This will be followed by a talk from Sonia, giving you some insight into the background behind her work and the research that she carries out. Following this, we'll have an in-conversation aspect, and please feel free to use the chat below because we'd like to open this up to the floor. We'd like to hear your questions and we'll weave these into the conversation. So yeah, without further ado, I will play the excerpt of the film. Thank you all for joining us today.
The mile-long causeway itself is a spectacle. It is lined with colossal concrete teeth designed to stop boats sliding through at high tide. The causeway is slick with seawater and the green of slippery, slimy, glossy and gleaming algae. The tide recently having receded to a secure remoteness. The crumbling, corroding, putrefying power of nature slowly creates entirely new, meaningful forms. Nature has now made it her own. The concrete ruin is consumed, engulfed, and appropriate creatures revel. The ruin is a constellation of meanings that move between past, present, and future. The ruin is a reminder of the universal reality of collapse and rot. A warning from the past about the destiny of our own or other civilizations. What constitutes life? What counts as a sentient being? What does creativity mean in the context of the laboratory? What does it mean to have agency and ownership over another's life? What is the role of human beings as makers? What do we think about the disposal of these lively objects that soon become infectious laboratory waste? What does it mean to be at risk with each other? What do we think of contamination? Entanglement is a fundamental aspect of all life. Yet we have more often than not been in common denial of these entanglements. But who are we? And what is ours? The I within us contains multitudes.
Hello, everyone. It's a real pleasure to be with all of you today. And my heartfelt thanks to both ASCUS Art and Science as well as Edinburgh Science Festival for hosting this event today. And without further delay, I'll just share my screen with all of you. So to tell you a little bit about the film, I hope some of you have already watched it, but for the benefit of those of you who still haven't had the opportunity to do so. So this film, in this film, I advocate for livable collaborations and rethinking bacteria as partners in health and survival of all living species. And the starting point for the film is a field visit to the ruins of Cremond Island. And this is a Scottish defense site from World War II, where I explored the manifold life forms that inhabit tidal zones. The film further investigates the potential of cohabitation and contamination for sustaining all life on Earth, especially in an era where we encounter many urgencies with accelerating rates of species extinctions, whereby a parallel can be drawn to the history of the wartime site, as well as the ongoing anthropogenic activities within the site that slowly become invaded by microbes that take back its space. So I asked through this film, how do political and economic activities come to shape the centers of our worlds, the tissues, fibers, forms of its lands and water bodies? How is the movement of non-humans remade through anthropogenic activities? And overall, it is my hope that cross-disciplinary collaborations might help us explore new ways of sensing, caring, noticing, and experiencing the world. The non-human touch has been realized in the framework of Entanglements of Time and Tide. And this is an ongoing research-based art science engagement project that explores the North Sea and its tidal zones in their ecological, cultural, political, economic, and poetic capacity. So I'm going to start with this aerial view of the island itself. And this is an image which is, this was taken in 2008. Across a causeway in the middle of the Firth of Forth lies the ghost island of Cremond. Cremond Island is a desolate tidal island about 1 mile out to sea, which is connected to the mainland at low tide across the Drum Sands. The liminal tide responding to Cremond. This causeway runs at the foot of a row of concrete pylons on one side of the causeway, which were constructed as an anti-boat boom during the World War II. At high tide, this path is immersed by several feet of seawater, which cuts the island off from the mainland. The mile-long causeway itself is a spectacle. The row of identical pylons evokes an imposing and arresting, if accidental, piece of brutalist architecture. The pylons are fissured, wrecked, dismembered, and fragmented here and there, where a pylon has collapsed and yielded to the waves. At high tide, these pillars are nearly submerged, and the landscape is at once wild, dynamic, and vulnerable. Scotland suffered more than 500 German air raids during the course of World War II. The country was a strategic interest point for Hitler due to its coast and many naval sites, one of them being the Scapa Flow in Orkney. Cremond Island, along with other islands on the Forth, was refortified and armed, designed specifically to tackle fast-moving torpedo boats. And so here you see the views of the anti-shipping barrier, which runs from Cremond to Cremond Island. And you can see the various phases in the construction of the pylons. And these images are from 1940. And here you could see the World War II era fortifications on the island itself. And there are various views of this. The causeway is slick with seawater and the green of slippery, slimy, glossy, and gleaming algae. The tide recently having receded to a secure remoteness. The crumbling, corroding, putrefying power of nature now slowly creates new, entirely meaningful forms. Nature has now made it her own. The concrete ruin is consumed by nature. It is engulfed, and appropriate creatures revel. Rising into ruin. The ruin is a constellation of meanings that moves between past, present, and future. It is a reminder of the universal reality of collapse and rot. a warning from the past about this destiny of our own or other civilizations, the very picture of economic hubris or industrial decline, a desolate playground in whose cracked and weed-infested precincts we have space to imagine or reimagine a future, a topography of insatiable greed and profit, which is easily forgotten in the midst of history's terrible repetitions. The ruin is a silent witness, and most unsettlingly perhaps, the ruin conjures up a future past, the memory of what might have been. We ask a great deal of ruins and divine a lot of sense and wisdom from their haunting silence. Everything comes to nothing. Everything perishes. Only time endures, no matter how fractured or dissolved at its edges. On Cremont Island, in the middle of the Firth of Forth, nature and culture, landscape and ruin begin to bleed into one another. We can no longer precisely tell what is ruin and what its circumstances, what is memorial and what the dead thing it recalls. The zone, a region, area, stretch of land, or section characterized by some distinctive feature, purpose, or quality, an area subject to particular political, military, or government function, use, or jurisdiction, a demilitarized zone, one of the divisions of the Earth's surface, an area, especially a belt of land, having a particular flora, fauna, determined by the prevailing environmental conditions, a portion of sphere between two parallel planes intersecting the sphere, a time zone, a girdle or belt, a sphere of thought, disagreement, and argument. Living material is collected from various environments of the zone. With time against us, we decided to retrace our steps and head back to the mainland. The tide was approaching swiftly, the waters of the Firth of Forth rising steadily. I felt that the decrepit state of things precisely reflected my own state of mind. I remained there for a while. It was cold out there in the causeway, with intermittent rain showers and a blustery wind. I was incapable of leaving, constricted like the water as it enters the drift of reeds, unhurriedly thickening and transformed. I look back at the mainland from the island. The edge of the sea is an enigmatic and extraordinary place. For no 2 consecutive days is the shoreline precisely the same. The tides progress and retreat in their timeless rhythms. The sea is not ever at rest, never at ease. I think about the life forms that inhabit tidal zones, far beyond just the human ones. I start to wonder, what does cohabitation mean in an era of many urgencies with accelerating rates of species extinctions? What is the association between capitalist devastation and collaborative survival within multi-species landscape? And what is the prerequisite for sustaining all life on Earth? What enigmas and nightmares lie beneath the palpable? Laboratory life constructing microcosms or microbial gardens. Back in the laboratory, we decided to construct devices for culturing a large diversity of microorganisms. And these are unique miniature microbial ecosystems or microbial gardens, as I like to call them, reusing the collected habitat. The structure of a microbial community is the result of environmental factors, evolutionary processes, and neutral or stochastic processes. Once prepared, the column is a self-sustaining enclosed ecosystem dependent only on input of light as an exogenous energy source. So much like a gardener tending to his plants and providing the finest conditions for a plant species to grow, a column provides a rich environment for microbes to grow or bloom as a lush population. And so these prepared columns were observed over several months for developments of layers, smells, colors, and zones. Observation 5 months later. As the microbes in the soil photosynthesize pigments, we are exposed to the processes of growth and decomposition of various species of bacteria within this ecosystem, with variations in population observed through waves of color. The aerobic as well as anaerobic gradients, along with the additive nutrients, allow for the growth and flourishing of various microorganisms such as chlorobium, chromatium, Beggiatoa, desulfovibrio, as well as rhodomicrobium. The water rapidly becomes anoxic towards the interface of the water and the mud. And there is still a capacity for the creation of biofilms as well as colony expansion. And then slowly, we also see a prominent green growth in the upper sections of some of the columns. What you're seeing is actually the middle sections right now, but you'll see that image later on in my talk. And this typical green growth denotes the presence of algae and other types of aerobic phototrophs. Culturing microbes, living materials and life processes as medium. Bacteria are a type of biological cell. They constitute a large domain of prokaryotic microorganisms, and they were the first forms to appear on Earth and are present in most of its habitats. Bacteria also live in symbiotic and parasitic relationships with plants and animals. What's interesting is that most bacteria have not been characterized, and only about 27% of the bacterial phyla have species that can be even grown in a laboratory. So bacteria were first observed by Anton van Leeuwenhoek in the late 17th century. But interestingly, they didn't become the objects of significant scientific study until the 19th century, when it became very apparent that some species causing severe human diseases. So the methodologies devised by Robert Koch, Louis Pasteur, and some of their associates during the golden age of microbiology are still extensively used today. During the middle of the 20th century, bacteria became widely accepted as subjects of empirical study in fields such as genetics, genetic engineering, and biochemistry. However, what's interesting is that today biologists face the conceptual challenge of rethinking bacteria as partners in health. In the Anthropocene, observing and perceiving microbial worlds is more essential than ever. Sustaining all life requires sustaining symbiosis, and perceiving these symbioses is a crucial way forward. According to animal physiologist and biochemist Professor Margaret McFall-Nye, human bodies can no longer be seen as fortresses to defend against microbial assault, but must be re-envisioned as nested ecosystem. And while elaborating on recent advances in the field of developmental biology, she says that new knowledge of the diversity and centrality of the microbial world promises to change the very face of this discipline, shaking its very foundations. And that our continuing goal is to define conserved processes governing the symbiotic associations, with the hope that we will provide fruitful directions for the study of more of these complex systems. Now, working with biological processes and structures can be both challenging and provocative. And creating and working with these lively materials, like I like to call them lively material, in the laboratory brings with it a range of ethical, social, cultural, and aesthetic inquiries, such as what constitutes life, who gets to determine what lives are created and grown, and which are saved, exploited, or destroyed. But also, some of the thoughts that crossed my mind were like, what do we think about using these living materials, such as living tissues, bacteria, and living organisms and life processes as medium? Or what does it mean to have agency and ownership over another's life? Do we think of this lively material as a carrier, holder, vessel, receptacle, or repository that is populated by our own thoughts and ideas? What is the role of human beings as makers? But then again, What do we think about the disposal of these lively objects that soon become infectious laboratory waste? What does it mean to be at risk with each other? What are the possibilities then and limitations as well of working with other than human lives? And finally, what do we think of contamination? So when does contamination become collaboration? So what you see are these jars, these microbial columns, and they are actually living systems where you see oil, sand, mud, air, seawater, biofilms, glass, additive nutrients, supplementation, including carbon and sulfur sources, as well as various microbes in various phases of growth and decay. And these microbes include cyanobacteria and algae, but also sugars and oxygen. The anthropologist Anna Lowenhoff-Zink says, and I quote, "The evolution of ourselves is already polluted by histories of encounter. They change who we are and make way for others. As contamination changes world-making projects, mutual worlds and new directions may emerge. We all carry a history of contamination. Purity is not an option. For staying alive for every species requires livable collaborations. And so collaboration essentially means working across differences. And this leads to contamination. Entanglement is a fundamental aspect of all life. Yet we have more often than not been in common denial of these critical interrelationships. It is imperative then to ask, who are we and what is ours? The I within us contains multitudes. The human microbiota, for instance, is the aggregate of microbes that reside on or within any of a number of human tissues and biofluids. So as humans, we are already colonized by many microorganisms, and those that live within us, the microbiome, are invaluable and irreplaceable parts of our lives. We are more microbial than human. In fact, we are less individual and more interconnected, interdependent multitudes. But do we acknowledge the significance of these symbiotic makings? Contamination makes multiplicity and diversity. Contaminated diversity is convoluted and humbling. And all these entanglements and symbiosis are vulnerable. And we must not ever forget that, because these relationships with others must be continually re-energized and negotiated within life's frameworks. When environs, times, and circumstances change, these very life-sustaining equations and balances become increasingly convoluted. Zink continues, if a rush of troubled stories is the best way to tell about contaminated diversity, then it is time to make that rush a part of our knowledge practices. And so what values emerge from these ruined landscapes? We are at risk with each other. Our becoming is a porous endeavor, an entangled world of unforeseen flows. It is in the wisdom of caring, in acts of mindfulness, and in the careful consideration of the discordance of disturbed stories that we may encounter our very best optimisms for precarious survival. Yeah, so I'll just quickly share some behind the scene images. And so this is me shooting at the site itself, and these are images from my field visit. And on the left side, you see the wonderful soil microbiologist and ASCUS lab technician Giri Girao, with whom I had the extreme pleasure of working with. And we've had some amazing conversations during these past years, not only during the residency itself, but also afterwards, you know, when we were sharing information as the microbes sort of flourished and grew in our columns. So it was really a very wonderful experience. And that's the ASCUS laboratory, which felt like home for several weeks. And I can't wait to be back there. And that's me looking through microscopes and really discovering these wonderful worlds. Another view of the ASCUS laboratory. And that summer hall where the ASCUS lab is situated. And that's once again, that's Jiri and myself working this time with microbiology. We were culturing these microbes at this time from the collected habitats. And so we're using the soil substrates and then diluting it through various measures. Another view of the ASCUS laboratory. And then just to highlight a little bit about the kind of conversations that went on, because this, the residency itself was in April, but like I mentioned, that we allowed these columns, these microbes to sort of grow and generate within these columns. And so we observed them all for over a year. And so there was this constant correspondence between Jiri and myself. And these are some of the images that Jiri had sent me. And this was the observation from July 2019, which was just a few months after we'd already constructed these columns. And here is another image. And I think I'd earlier spoken about the green growth on the top phase. And I'd mentioned that, you know, I would be showing this image later on. And so this green growth that you can see on in the central image, as well as the one on the right. And so this is attributed to these phototrophs. And so there's also algae and cyanobacteria.
Great, thank you, Sonia. Yeah, it's really heartwarming to see those images. It feels like many moons ago, and Jiri is no longer in ASCUS lab. He's back in Czech Republic. But yes, yeah, it was great to see the kind of work and the conversations that emerged from that one intensive week and the field trip to Cramond. So yeah, it's lovely to see those behind the scenes as well from your perspective.
Absolutely. But also the kind of vulnerabilities, right? With this whole process where the artist is working, but also throws open these kind of these ethical questions as well, related to the kind of research that is happening. And so I found this a space which was very enriching for me on so many levels. But of course there were answers, but there were also significant other queries that sort of surfaced, and I'm actually really thankful for this experience because I found that, that I found like I was growing on so many levels, right?
And what, in terms of the lab, have you worked intensively in a lab like that as part of your creative practice, really delving into those processes? Because I know yourself and Jiri, you did culturing together, you made the Vinogradsky columns together, you went on a field visit to Cramond together and collected the samples. I remember we had a conversation, even things like sample collection. I think when you were in Aberdeen, we were asking, oh, could you bring some samples from Aberdeen? And you were like, what do I do? How do I do it? What's the way? And we were like, it's simply just take a bottle, collect something. But it's funny, the language of collect a sample. If you just ask someone to collect something, or you collect a sample, it makes it sound like it's something way more complicated than it actually is. But yeah, how did the process of being so physically immersed in the lab kind of impact on your practice?
So this is interesting that you bring this up, because the first time that this question also corresponds to another question that could be raised at this point about when I started looking at art science collaborative projects, really. So it sort of ties in. And I look back, I think about the very first science research residency that had enabled me to see this wonderful world. And this was back in 2015. And this was a research residency at the MSSRF, and that's the MS Swaminathan Research Foundation in Chennai, in India. And really, that was the very first time that I was introduced to this world of lab-based practice. And however, there are differences, because at the time, I worked with scientists, but of course, the culturing of the bacteria, for instance, I worked with a lot of bacterial samples from the mangrove ecosystems. and also the coastal ecosystems around India's Coromandel Coast. But of course, having said that, because I don't have a background in science, the scientists themselves were really doing the culturing. And so my role was to have these discussions with them, intensive discussions, go on field visits with them where they collected the samples and then they cultured the microbes as well. And so a lot of my process was about this kind of correspondence with them, communication with them, sharing of ideas, but also a very intensive documentation process that followed. So what was different with the ASCUS residency was that this all became very hands-on for me for the very first time. So the very first time I actually had the opportunity to culture these microbes myself in a laboratory. But also, I had a far more basic understanding of how a microscope can be used before this. And so through this residency, this sort of opened up this whole window of opportunity for me. And I was able to study and learn so much alongside. And so I think that this is something that stays with me for life. But like I said, doing this kind of culturing and this kind of this lab-based practice itself brings with it all these range of questions. And so I've actually gone ahead and so bared my soul in this video where I've actually talked about some of these vulnerabilities and what kind, as an artist, what are the kind of dilemmas that one really encounters when you work with this kind of lively material in the laboratory where you actually, you are actually growing these microbes. So on one level, they're like progeny, right? And then you come to a phase where you actually monitored, you've grown them, you've created them, and then they at the end of your so-called project or whatever it is, they are infectious laboratory waste, and there's a certain process through which they must be disposed of. So those kind of questions, again, they're all in the gray, right? Because there is no very easy way to sort of either understand these or to sort of communicate these experiences. Some of them are really very deeply personal experiences as well, but I've tried to do some of that quite honestly in this film and through my work.
Yeah, and I think we've seen that, we have other artists that we've worked with in the lab and kind of working with, as you call it, this lively material just does kind of start to change your perspective of that relationship that you have to this material.
The sensory experience itself, right? The touch, the experience of the smell, the touch, so on and so forth. And that's where I think it's really made a huge difference, and it's really been a life-changing experience for me going forward. Yes.
Absolutely. And we have a question just relating to that from one of our audience members. So they've asked, is any of the lively material specific to the site and its entanglement?
Okay, so all the lively material has been made or created from this reused habitat of Tremont Island. So everything that you see in the film in terms of the columns themselves, those images that you saw of the bacterial growth, but also of the petri dishes, the cultures themselves. And so this is all been created from reused habitat. So that's like water samples, soil, mud samples, but then we also used biofilms, from these pools of water between the pillars themselves. And so they look really interesting. And so we thought that, Jiri also informed me that those are kind of sites where you see a flourishing of microbes as well. And so we collected those as well. And so all of this work for the film is really, all of it corresponds to the site. And now I think the question of entanglements is really interesting, because, like I have talked about the wartime history of the site. And so the reason for doing that is pretty much to talk about these, when you first encounter the site, that it's not largely inhabited, but it is largely uninhabited. And so also, and so you presume that the site is pristine, and it's uninhabited. So there's a certain impression you have of it at once. But then you do some research, and then you you come to know that, this site has had a really long history of anthropogenic activities. And that's really interesting because although I have talked about this from the World War II period onwards, the human connection with Cramond Island and the Firth of Forth actually goes back apparently to prehistoric times. And so there's a burial site there in Cramond somewhere, which was then discovered much later. So it begins with that, and then it moves on to the Roman period. Although if I'm correct, then the Roman foothold in Scotland was really not very strong, but Cramond is one of the archaeologically rich sites that you would find. And so there's a Roman outpost, which is somewhere near the island. And so it is presumed also that this island could possibly have been used by them in the past. But then moving on, again, the site had a big role to play during the World War I. And I think this was probably in 1914, sorry. And so this was a defense site once again during the World War II, and then in World War I, and then in World War II, it was refortified and armed once again. And then again, I think in 1939, if I'm not mistaken, there was a whole episode of air raids, not on the causeway, but close by near the Forth Bridge. And this was actually, it's interesting because this was the very first air attack by the British on these lands. And this happened over the Forth Bridge. So really, if you look at past activity and our entanglements, it goes as far back as the prehistoric time. Although at first impression, you wouldn't think so. But again, if I talk about more recent developments and through what I found through, and you will see this in the film, I haven't used those excerpts for my presentation, but if you see the film, you'll see that there are the excerpts of reports, science excerpts from science journals, but also from, reports that were published at the Scotsman, by the Scotsman, for instance, which establishes the connection of this site with heavy water pollution and as well as bacterial growth. But also there is this immense amount of domestic and industrial sewage, which is untreated, which flows regularly into the Forth. And because of this, the shellfish that you find in this area, this is contaminated to a very great extent. And so there have been instances in the past where there has been, the police as well as, the environmental officers, they have collected over 100 kilos of contaminated shellfish that have been infected by very dangerous bacteria. And so then you can imagine that the possibility of this infecting humans as well, for instance, right? So it is all a result of our own engagement and our own entanglement with this site, even with what seems to be a remote site. For instance.
Yeah. And so you kind of, I guess this is the kind of how the historical context kind of plays a role
Absolutely.
Within the framing of the work. But it's you mentioned there these words and they're kind of, they come through in your work at different points: contamination and toxicity and ruin. And these obviously have negative connotations. So how do you think we reframe our relationship with microorganisms to reconsider kind of the meaning of these words?
Yeah, I think that's a really interesting question, Miriam. And I think in today's time, in these very urgent times, I think if we look forward, there is really no way for us to live other than to live with the toxicity and contamination that we have created around us. And the pandemic itself is the greatest example of that. And so I think the question that we're all confronted with at this point is that how do we positively, or is there a way to positively think about contamination going forward? And so if we look at the idea of contamination, toxicity, and entanglement, and we start to look at ourselves, like I've said this in, even in my presentation, but if you look at the idea of the human body being not from the perspective of an I, but rather a perspective of a we, that, and to also look at our system as a multitude, something that harbors the bacteria or the virus is already within us. So this kind of a change of perspective, I think, could be a very interesting way to look forward. And so also, I think the other thing also to think about is that, even if you look at scientific research, you have this, we've always been used to, since our childhood, looking at this tree of life, and we are all familiar with this, but this tree of life itself, the idea and conception of this tree of life, where the human is at the top and therefore presumed to be the superior being, with the microbes at the bottom of the list. This hierarchical positioning, I think this is the thing that needs to be questioned. This needs to be questioned. This needs to be understood more thoroughly. And so these are the kind of questions that are coming up not only within the scientific field itself, but also within the cultural communities, within the artistic arena as well. And so that's where I feel that these these interdisciplinary collaborations have such a role, such an important role to play going forward, because I think we need the strengths of both the languages today. And again, I mean, just if you look at everything that has happened in the last year and a half, look what the microbe has taught us. And, for instance, so if we look at ourselves as superior being, we really need to reconsider that, because clearly it is the human being that is the most vulnerable, because life will go on without us, but and organisms will continue to flourish. And so this is a very, very important lesson already. Absolutely.
Yeah. And I know in relation to this, kind of, I guess because you started working with these scientific subjects and weaving these into your work and with microorganisms a number of years ago, from your perspective, has working with this lively material, has this changed your own perspective? You talk about the I versus the we in these nested ecosystems and the microbiome. So how has when did this change or shift happen for you, or how did it happen?
Well, again, if I go back to looking at some of my earlier projects, and critical membrane has been one of the projects that I've been working on. It's nearly been 8 years that I've been working on this project in the southern part of India. And that project is still ongoing, as are many of my projects, because I like the idea of engaging with a site for a really long time. And therefore also I find myself observing the changes that happen, in these sites gradually and over a period of time. But again, working with microbes and with bacteria in the laboratory back at MSSRF has also taught me a lot, because for one thing, I started to look at both the ethical as well as the ethical and the political angles which are associated with any kind of scientific research or conversations. And so I think it's very important to say that, that we essentially, humans are political and social beings. And so the political drives everything around us. And so I think for us to be alive at this moment, it calls upon a certain requirement or a certain necessity for us to respond to the crisis in several ways and in various dimensions. And so so this is the kind of thing that I've been thinking of for a very long time. And also the whole idea that, like if I look back at my own life and my personal experience of pregnancy, for instance, the whole idea that the fetus is actually a foreign body that comes into the mother. And so this is the immune system's very first response would be to eject a foreign body. And it's so interesting that, on the contrary, the microbes in your system are actually your own, and they work for you. And therefore, I mean, it's a symbiotic and collaborative process, because if the microbes are healthy in your system, in your body, in your gut, or wherever else, you're healthy. If they're eating well, you're healthy, you feel yourself, you feel energetic and things like that. And so that's the other thing to think about, like this kind of this mutualism and this symbiosis. And so how do you think from within, really? Like first you think from within, and if you're able to do that, and then I think the experience of then you maybe you have a chance of experiencing the world in a different way and looking at other life forms in a different way.
Absolutely. And you talk about microbial gardens and, as you mentioned, these different ecosystems, the ones both within our body and outside. Yeah. This is quite an evocative term. How is this framed within your work?
Yeah, so essentially, when I would say that at least for me, when I look at a garden, I think of the garden as two things. I think of if I look at my own garden, so I think of two things. I think that this is a work of art, but it's also, my garden is a work of science. And the other thing that one does for the garden regularly is to care for your garden, to really sense and experience your garden, but also to tend to it regularly. So I think, if you reverse things and you look within yourself, the idea that, your body itself, the interior, is also something that you take care of, you must take care of, and self-care, I think, is really important. And this is something that's been spoken a lot in this time of the pandemic, that how first, you need to take care of yourself, and then you think of, how you might extend that idea of care and nurturing outside of yourself. And so the garden, I think, is a great symbol. And this whole ideas of tending, experiencing, but also just experiencing and sensing. Yes.
So the changes that happen to our kind of life-sustaining ecosystems as a result of human behavior are also very central to your work. This idea of our impact on these ecosystems around us. So why is this focus so important to your work?
I think being alive at this time calls for us to really definitely take action, because there's a certain responsibility I see going forward. And this is why I also feel that interdisciplinary collaborations are really so essential at this point. And therefore, if I look at my own collaboration, I feel like the voices of the art, the voice of the artist, the voice of the scientists, coming together through my work, I think this is really very, very, very important. Not so, in this particular video, there isn't really a conversation with the scientists themselves, but in a lot of my other works, some of them which are still are already in production, there are engaging conversations with the scientists themselves, but also the scientists being the center of the focus, himself or herself. So I think it's important to hear the voices of both. And here I would also, people often ask me about this whole idea of art and activism. Do you see yourself as an activist? And this kind of a question comes up very often. And I would just like to say that, we should, at this point, we need to have, what we really need is to have a little bit a broader understanding of the word activism, a more inclusive understanding of the word activism. And it's really fantastic for people, for those of you, or for those of the people, or the people who are engaged on field, for instance, engaged on site and working with communities and things like that as well, but also the people who lend their voices to protests and things like that. But also just to consider how an artist's personal practice can be a form of activism. I think for me that is extremely important. And also how within your, you have these stories to tell, and what kind of stories you have to share within your personal space. And so extending this idea of activism as a sense of growth, on every level, I think this is extremely important. And so as artists, we are receptive and we are empathetic. Empathy, I feel, is very, very vital for us today. And this is the language of empathy, the language of love, which is really, really most important. And so we feel things deeply. And so it's from the depth of this feeling that we really respond. And our work is a part of that process of activism itself. So really, it's really that.
Yeah, and activism, kind of, it exists across different spheres in different ways, and there can be silent activism and other ways to be part of the change that needs to happen. And I think we do all need to work together and find our voice, as being that activist, but in different ways. And I agree, kind of, there's a the reclaiming of, I guess, what is an activist, I think is something to consider.
I have a question for you, Miriam, actually.
Yes.
You know, like it's interesting. So you you've been working with artists for a long time, but also what are some of the questions that come up as a part of their discussions when they are involved in lab-based practices? So speaking from the perspective of some of the others as well, you know, who've been a part of this process and who've had the experience of either being scientists themselves and working on a project at the laboratory, but also the experience of other artists from across the globe that have worked together on projects with ASCUS and interacted with some of your most wonderful lab technicians and scientists.
Obviously, artists do ask questions, and what we do is we try to invite the weirdest, most wonderful of questions. So there's no question too strange for us. And I guess what we see ourselves more as the facilitators and the support network, and that physical space to really indulge those artist questions and invite them to explore and help them to meander along, I guess, the path that they find themselves on as those lab processes open up their understanding a little bit more, because I guess it's quite a unique space and artists don't necessarily have easy access to the types of facilities that we have, and maybe haven't done, maybe like yourself, have seen culturing happening take place, but haven't actually done it themselves. And so I guess, and maybe you can give more insight to this, I guess, as going through the process of culturing, and as you do that and you become more familiar with it, even from my perspective, having done culturing as part of the lab, just setting up the facility and being involved in it, there is such an art to culturing, and it's a bit like cooking. You have to feel it, and it's always, the lab technician will be like, you just do this, and I was like, I understand the instructions and I understand what I need to do, but I don't know what I'm looking for or feeling for, because it's that subtlety of, even on a practical level, how the pressure that you put on the agar jelly when you do the culture.
Absolutely. There are these nuances and these intricacies.
Mm-hmm. And when
that
something
are really
grows,
so important.
And when something grows or doesn't grow, I ask, why didn't it grow? Why did it grow? Was it something I did? Was it something else? And biology itself it does what it wants to do. You can try to coax it and encourage it and want it and will it to do something, and sometimes it will, but sometimes you repeat the exact same thing and it just doesn't.
So I have this brings me to a very interesting point, and I remember my conversations during the residency with Jiri. And I remember being so curious and asking him, we've made these columns and everything. Do you think that anything will grow? Will we, what will happen? And at this point, I said, I'm so curious because, there's also this incubating time, and there's this waiting window, where you allow the microorganisms to sort of do their thing as well. But also, so he said that, we don't know, we really don't know if we are going to get any result, what you might have in mind, and what you get, is could be something, two completely different things. This could be. So this experience of also the not knowing.
Yeah. Absolutely.
This is a very much a part of the experience itself.
Yeah, and I think that's often as part of the work that we do, that the managing of expectations. The irony is when you run workshops or activities that involve scientific experiments, and at the end you've kind of go, well, you've got to wait because it needs time to grow, so you won't really see anything yet. But when you come back, and then also it might not work, but that's okay. That's normal. It's not like you did anything wrong.
It's a part of the process.
It's a part of the process. Yeah. Exactly.
If you'd like to just quickly mention the Entanglements of Time and Tide project that's coming up, and how these objects are going to be a part of the exhibition for audiences to come experience and see for themselves these objects these lively objects that we created. but also how they are also going to continue to grow, flourish, decay during the couple of months that the project will be on view, nearly 5 months for 5 months.
So we'll be working with Edinburgh printmakers to see what living aspects will become part of that physical exhibition as part of the Edinburgh Art Festival. So at the moment they have an online viewing room, but this is kind of the digital snapshot of what will be the full physical exhibition, which launches on the 30th of July, isn't it? Yes.
Yeah, that's right.
So please do keep an eye out for that coming along. Please do go online and watch the full film. The Non-Human Touch is available on our website for the duration of the Science Festival. So do check it out. And also we're running a follow-up event this coming Friday, and we're delighted to be joined by Professor Colin Moffat, who's the former Chief Scientific Advisor to the Scottish Government for Marine Science. So it'll be amazing to have him. He's just recently retired. So he has just has some fantastic insight on marine ecosystems from the scientific perspective. So do book onto that. There's still plenty of spaces available. But yes, I think that probably wraps us up for today. I'd like to thank you, our audience, for joining us. I'd like to thank you, Sonia. We're delighted to finally bring this work into the public arena for the Science Festival this year. But we will see you again on Friday.
Thank you, everyone, for your time and for your patience. Thanks for joining us.